today's speaker, Dr. Yarimar Carrasquillo. Dr. Carrasquillo is the principal investigator at the National Center for Complementary and Integrative Health at the National Institute of Health. And she directs a multidisciplinary research program aimed at delineating the mechanisms underlying the bidirectional modulation of pain in the brain, with a focus on the central amygdala. Dr. Carrasquillo was born and raised in Puerto Rico, and she is a proud alumni of our institution at the Recinto de Rio Piedras. In fact, we have in the audience her mentor, Dr. Sandra Peña de Ortiz, who was an undergraduate, and I was also in Sandra's lab when was one of the first undergraduates that we trained together. And Yarimar, in particular, I always remember how inquisitive and bright and so responsible. So she was kind of the role model, right, Sandra, for our students.
Yes.
In particular, and we're very proud to see what she had achieved through her career. So when she finished her bachelor's degree with us, she moved to Texas to obtain her PhD in neuroscience at Baylor College of Medicine in the lab of Dr. Roger Giraud, propelling her career in pain research. She focused her postdoctoral training in the cellular physiology in the lab of Jean Nerborn at Washington University School of Medicine, and then she joined NIH as a tenure-track investigator since 2014. In addition to being passionate about pain research, Yarimar is very strongly committed to increasing diversity and inclusion in science. So today she's going to talk about the neural circuits for the modulation of pain analgesia in the amygdala. And I would like to mention that we'll have her give her presentation, and then we'll have a Q&A session. So please include your questions through the chat, and we'll be able to have Dr. Carrasquillo respond to them. So without further ado, Yarimar, thank you so much for accepting our invitation, and we'll hear your wonderful science. Thank you.
Well, thank you, Dr. Maldonado, for the invitation, the kind introduction. My heart is racing. I'm so excited and happy to, even if it's just virtually, just the fact that I'm back home, and that having Sandra in the audience and Dr. Maldonado, like it's pretty surreal. So I'm very much looking forward to sharing what I've been doing. So as many of you in the audience, I started my career by being interested in the brain mechanisms that underlie behavioral responses. So I've always been very curious to know what is happening in the brain that drives so different behavioral outputs, both in adaptive and maladaptive responses. And like many of you in the audience, my journey began at the University of Puerto Rico as an undergraduate, as Dr. Maldonado mentioned in the introduction. This is us in my first neuroscience meeting that Dr. Peña welcomed me into her lab and just introduced me to the world of neuroscience. And I joined her lab in 1997. I was in second year of undergraduate. In Peña's lab, we were interested in understanding the molecular mechanisms of learning and memory. And it got my attention because, again, I was highly interested in knowing what was going on in the brain in the context of behavior. And when I first met Peña and just looking at the title of the lab, I thought that it would be like ideal to begin my journey. And I had zero experience in neuroscience, and we used long-event rats, and we used the whole board food search task as a model for learning and memory, spatial learning and memory. So this consisted of sort of like an open field that had 16 holes. And what the rats had to do is that I tried to put little drawings, but my lab vetoed it, so I took them out. But in the room, we had spatial cues in the walls And the rat would enter through this door, and then using the spatial cues, we would assign a specific pattern of 4 holes that had food, little Fruit Loops, I remember. And the rats would really want to go in. They were a little bit food deprived, but not a lot. And their task was to learn how to navigate the maze using the cues so that they could learn where the reward was, the food pellets, right? So we measured the learning experience by looking at the time that it took the animals. We did this for 5 trials a day for 5 consecutive days. And we look at the time that the animals took to find all 4 of the Fruit Loops, and also the errors that they committed. And as you can see, which were defined as visits to empty holes that didn't contain the little Fruit Loop. So as you can see here, it was very clear that the rats were learning the task very quickly in the period of time that we looked at, therefore validating that this was a very useful model to study learning and memory. If we remove the cues from the walls, then the animals had a lot harder time finding or identifying where the path was. So as a fresh undergrad, just seeing, I will never, ever forget this experience, because I remember just how impressive it was to me that such a tiny animal that I didn't know could be so smart could learn so quickly to do this very seemingly difficult task. Now, what we did is that we used that task to then ask the question of what's happening in the brain. And to do that, we took out the brain of the animals and then look at the mRNA level, looking at a specific immediate early gene called HCF3, in order to get an idea of whether there were changes associated in this immediate early gene in the context of learning and memory. When we did that, another first and a big surprise for me was that we could see when we took the brains and we sliced them, we could see that what you're seeing here is the hippocampus. And in naive animals that were not exposed to the maze, there was very little expression shown in gray of the mRNA for HCF3, but in animals that were on day 3, so about here, that they have already learned the task, we saw a very big upregulation of the mRNA for HCF3. And the reason why I'm showing this is because this experience as an undergrad was a very defining moment for me in my career. It taught me, one, that I absolutely love behavioral neuroscience and that's what I wanted to pursue in my career, but also that there were these fascinating tools that would allow me to go all the way from the animals and visualizing changes in the brain, which I didn't know from just basic biology courses. With the encouragement, support, and mentorship of both Dr. Peña and Dr. Maldonado, I moved on to continue my career. And fast forward 20 years, and I'm still asking the same question, which is, what are the brain mechanisms that underlie behavior? But now I am interested particularly in pain. And the reason for this is because when I moved to Baylor, when I was interviewing, I learned that while I was very focused on the good plasticity associated with learning and memory, plasticity could also be a maladaptive change that, if it went bad, it could also underlie pathological states such as pain. I was drawn by the similarities in the cellular and molecular mechanisms between pain and learning and memory, and decided to move forward to that field, which I've pretty much stayed. So basically 20 years go by and I'm still with the same sort of big question, focused on pain, but now I'm at the NIH with my own lab and addressing these questions with my really amazing group. Now, what do we know about pain? We typically think that pain is a bad thing, right? But it's very important to know that pain has both a good kind of pain and a bad kind of pain. The good kind of pain is that we need pain in order to protect us and as a warning signal. The bad thing is when pain just takes over and doesn't have any protective role. Now, we also know that pain is not static. It can be modulated up and down by many different factors. And the typical examples that people think about is that pain can go down during stress-induced situations, like the typical one that people think about is soldiers in combat, that they get injured and don't even notice that they're injured. But then pain can also go up in maladaptive cases, such as low back pain, neuropathic pain, inflammatory pain, arthritis, you name it. You know many of them. Now, for a very long time, we've known that the brain has the ability of turning pain up and down. However, the mechanisms that drive this dual function of the brain in the modulation of pain are not clearly elucidated. So with this in mind, then I decided to focus my research program at the NIH with the specific goal of identifying the mechanisms that underlie the bidirectional modulation of pain in the brain. And as Dr. Maldonado said in the introduction, we use multidisciplinary approaches. I like to always start with the whole animal. In this case, everything in our lab is now in mice. And just like I learned in Dr. Peña's lab, we start by developing a model that mimics what we want to study. In this case, is pain. And then, again, similar to Peña and what I also did as a grad student, then we do two different types of measure experiments in the lab. One is to look histologically at whether there are changes at the anatomical and histological level that are associated with the pain responses. And we also go and look at the changes at the cellular level. This was expertise that I gained as a postdoc, where we look at, if we patch neurons, I always say this is like zooming in a lot. So if we go and patch individual neurons, what are the changes that are, again, associated with the behavioral models? Now, once we have these measure approaches, now what we always like to do is to go back to the animal and manipulate these events pharmacologically or with different tools that you will see, and then test causality of the different hypothesized mechanisms that we're seeing in the measure experiments. So we focus our work on the central amygdala, and the main reason for that is because, again, I was interested in the brain, so I wanted to look at a brain structure that was well positioned to modulate pain. And I selected to focus on the amygdala because it receives direct nociceptive inputs via the spinopontoamygdaloid pathway, but it's also a very important site for the modulation of affective behaviors and emotions. And we know that the International Association for Pain defines pain as an emotional reaction. It's a negative reaction to a noxious stimuli. So with that in mind, then we focused on the central amygdala, and my graduate work, 2000 to 2005. Now this feels like a historical talk. It demonstrated that the central amygdala functions has a pronociceptive role, meaning that it promotes hypersensitivity, pain and fear, pain and affective comorbidities. Our work demonstrated in grad school, my work demonstrated that the central amygdala had endogenous molecular mechanisms that drive the hypersensitivity. And there's been a lot of work in the last 15, 20 years that have really demonstrated and capitalized in the different tools that we have to unambiguously demonstrate that the central amygdala is pronoceptive and has therefore even been referred to as the nociceptive amygdala. Now, when I joined Rob Lab as a grad student and we were looking and thinking of studying the central amygdala in pain, I remember that it was not so black and white to think that the central amygdala was going to promote pain. And the reason for that is because studies in the 80s and 90s had demonstrated the complete opposite function of the amygdala. So manipulations that now we know interfere with pronoception, same manipulations performed by labs that are interested in studying analgesia, those labs demonstrated that the central amygdala was also anti-nociceptive. And I tend to get obsessed with things, and I moved on with postdoc and with different things, but this has always bothered me, the idea that there are two different lines of work that are showing completely different functions. So at the time that I started my lab at the NIH, what I proposed is that maybe it's not that these labs are right or these labs are wrong or the other way around, but that possibly the central amygdala had actually a dual and opposite function in the modulation of pain, so that it could do both. Now, how is it doing that? So the task number one is to test the hypothesis that it actually has two functions. And then the second most important question for me, because I've always been driven about mechanisms, is how is it doing this? So we knew that all the cells in the central amygdala are GABAergic. So that in itself was puzzling, because one cannot say that the opposite function in analgesia or hyperalgesia are because of recruitment of different excitatory or inhibitory cells. We knew that they were all GABAergic. But right when I was starting my job search and when I was building my research program, it was very clear that the central amygdala, in spite of the fact that they were all GABAergic, it became very clear because of the different tools that the central amygdala is very heterogeneous. So it has neurons that have a lot of heterogeneity in terms of morphology, electrophysiology, and their genetic identity. So we knew from work that had been published quite recently at the time that there were two major cell types defined by their genetic identity. The cells that express somatostatin, which I'm showing here in red, and the cells that express PKC delta, which I'm showing in cyan. Now, these two populations constitute most of the central amygdala, but the nice thing about it is that they're non-overlapping. So I decided to focus on those two because it would allow me to study two independent populations that are intermingled within the central amygdala, but are separated in terms of co-expression. The specific hypothesis that I had is that modulation of pain in the CEA was going to be both cell type and circuit specific. Now, what I'll show you today is the work that we did to demonstrate, well, the first is to test the hypothesis that pain-related behaviors are cell type specific, looking at the PKC delta and somatostatin cells. Then I will show you work in the lab. We always like to take the airplane view and then zoom in and look at the electrophysiology. So once we characterize the function of these two cells, then we wanted to dive into the cellular and morphological characterization to see if those cells are different electrophysiologically and morphologically. Now, we go back to the airplane view, and now we want to know also whether the inputs that are coming, where are the inputs coming into the central amygdala. And then lastly, I will show you the data that we have gathered demonstrating or aiming at looking at cell type specific projections to pain-related behaviors. So for most of the experiments that I'll show you, we use the cough model of neuropathic pain. So basically, we do several different test, but this one is for tactile. And what we do is that we have these nylon filaments that are calibrated, and when you press them against the paw, they exert a specific amount of force. We poke the animal with increasing force. When the animal withdraws, we know that that's the threshold, and we call that the threshold. Once we establish the baseline, then we go in and we expose the sciatic nerve, and we put a little plastic tubing around the sciatic nerve, and that will make the animals feel hypersensitive, similar to when people have like a pinched nerve or herniated disc or things like that. We then repeat the bone fry test, and the idea is that now the animal will tolerate way less force and it will be withdrawing. The graph that you will see will be similar to this. This is the paw withdrawal threshold. In other words, the force that is required for the animal to withdraw the paw. And before the surgery, all the animals had similar thresholds. But then after we insert the cuff into the sciatic nerve, we see a very drastic reduction in the threshold, so that now the animals can tolerate very, very little force, and they're already withdrawing the paw. I like this cheat sheet so that you can see that in all the manipulations, you're going to have this little cue that tells you in which direction the modulation is happening. Now, in order to look at the specific cells, we're using the Krylov system. And just to give you a basic idea of how this works, we use different Kry lines of mice that basically will have, under the promoter of either PKC delta or somatostatin, Kry is under these promoters. And what that means is that any cell in these animals, any cell that expresses PKC delta will express Kry. Any cell that expresses somatostatin will express Kry. Now, in itself, Kry doesn't do anything. So this mouse doesn't do anything, is pretty much identical to wild type. When we cross this animal with a reporter animal, and that reporter animal has the ability to express a fluorescent protein, red TD tomato, but it has a stop sign, a molecular stop sign, that doesn't allow the animal to have TD tomato unless Kry is present. When Kry is present, the enzyme will cut the stop sign, and now the animal will be able to, every cell that has PKC delta will be red, or every cell in the somatostatin Kry animals, every cell that expresses somatostatin will be red. Now, using these animals and a similar approach to what I did in Peña's lab for the HCF3, except that now we're looking at a plasticity marker called ERK. What we do is that after we induce pain in the Kry animals with the reporter, we take out the brain and we take slices of the CA and we look at the plasticity marker ERK. So what you're seeing here is in cyan is the ERK marker. You can see that there's a very robust ERK activation in the central amygdala when we induce the pain. But when we look at the PKC delta cells, the colocalization, because we wanted to know which are the cells that are being recruited, what we see is that most of the cells are localized to the PKC delta cells, most of the ERK positive cells are localized to the PKC delta cells. So you can see here that pretty much everyone is colocalized, and you can see it also in the low magnification image. And these are the quantification. When we look at the somatostatin expressing neurons, it's virtually no colocalization. This was the very first finding from our lab, and it was really exciting because we were like, jackpot, we have the pronociceptive cells. It has to be the PKC delta expressing cells. Now, as you all know, these molecular markers are fantastic, but they are still indirect ways of measuring activity. And as I learned from my postdoc mentor, Jean Nerbonne, she says electrophysiology never lies. So I always like to go back and look at electrophysiologically what we're seeing with the behavior of the cells. And in this case, we use slice electrophysiology, so very similar, the same animals, but now instead of fixing the brain and looking histologically, we keep the slices alive. We can see the fluorescent cells, we patch them, and we record the activity in the form of action potentials. When we inject depolarizing current, the more injection we inject, the more current we inject, the more action potentials we get, as can be seen here with the increasing current injection, increased number of spikes. I like this a lot because it's a cellular correlate of what we see with the behavior, so that we see that with increasing force, we get more responses, right? So this is similarly, it's like a cellular correlate of that. So what we saw is that with the PKC delta animal, consistent with the ERK activation that we saw, in the control animals have much less action potentials than the pain animals. So we have increases in the excitability consistent with the ERK activation that we saw. Everything suggesting that these cells, that activity in these cells contribute to hypersensitivity. Now, in the next set of experiments, we wanted to know if the activity of these cells is actually necessary, so to try to establish a causality. So now we go back to the animal, and we use a very similar trick to what I showed you before, but now instead of crossing the mouse with a reporter mice, what we do is we go and acutely inject in the central amygdala a Cre-dependent modified G-protein coupled receptor called a DREAD, and it's inhibitory. Just like with the other, just like with the reporter, the DREAD in itself is not going to do anything unless Cre is present. So what this does is that it will target the the DREAD expression into specifically the PKC delta expressing cells. We always like to validate everything that the approaches that we're doing. So when we patch these neurons, we can see that, oh, and the DREADs are G-protein coupled receptors that in itself don't do anything. They're activated by CNO. So when we apply CNO, we can see that the inhibitory DREAD does what it's supposed to be doing, which is to inhibit the cells and prevent firing. The M-Cherry control didn't have any effect. Now, using this approach, we go back to the animal to do behavior. We put the inhibitory DREAD. We do a very similar battery of behavior test, and we measure the responses of the animals with or without CNO, which, remember, what it does is it blocks the activity of the PKC delta expressing cells. When we do that, what we see is that in all the control conditions, the cough animals are hypersensitive, but when we inhibit the activity of the PKC delta expressing cells, we see a very robust reversal of the hypersensitivity, so that now the animals, in spite of having the cough in there, they don't feel the pain anymore. It's basically back to normal. If we come back the next day, the animals are still hypersensitive. So this is very specific to the acute manipulation of the activity of the PKC delta cells. Importantly, we don't see any effect in the sham animals, demonstrating that this is an anti-hyperalgesic effect and not just that it's bringing the threshold up, kind of like the way that lidocaine does when you apply it to the skin, that you just stop feeling anything. This is just blocking the hypersensitivity induced by the cough. So, whoops. So now that we have shown that activity in these cells is necessary for hypersensitivity, in the next set of experiments, we wanted to see if we could activate the cells, if that would be sufficient to induce hypersensitivity. So to do that, we use a different flavor of DREADs. Basically, it's a very similar experiment, but now this modified G-protein coupled receptor, when we put CNO, instead of inhibiting the cells, it activates the cells. So when we activate the PKC delta cells, what we see is that now the animals that were typically not having any pain, now they behave as if they have the cough, but these animals have absolutely no injury or no manipulation other than the activity of these cells. So clearly demonstrating that activity of the PKC delta cells is both necessary and sufficient to induce hypersensitivity peripherally. Now, it always bothered me that what are the somatostatin cells doing? All that we knew is that they don't express ERK, and also the immediate early gene CFOS, we didn't see anything, no colocalization with the somatostatin cells. But as a trained electrophysiologist, I came back to my slice data, and I wanted to see what is happening electrophysiologically to the somatostatin cells. And when we started to patch these cells, the very first thing I noticed is that the pattern was quite different to what we see in the PKC delta cells. So we have a lot of spontaneous cells that are just firing all the time. It's about 50% in the control animals, and about 45% are quiescent at rest. Now, to our surprise, when we implant the cuff, what we see is that now most of the cells are silent at rest. So we barely see any cell that is spontaneously active. So this is demonstrating that in the somatostatin cells, cuff implantation silences the neurons, which is completely opposite to what we saw with the PKC delta neurons, that we saw increases in the firing responses. So this suggested that possibly these neurons were doing opposite functions in the modulation of pain. So now we're going to do a similar set of experiments to what we did in the PKC delta cells, but this will be in the somatostatin cells. So when we block the activity of the somatostatin cells, which would be mimicking to what we see in the context of pain, what we see is that now the animals behave as if they had pain. So these are the sham animals, in spite of not having any injury. This, as a reminder, is the opposite to what we see in the PKC delta animals, that when we inhibit, we see a reversal of hypersensitivity. We next wanted to know, can we reverse the hypersensitivity if we increase the activity of these cells the way that they normally behave when there's no maladaptive plasticity? So we use the GQ dreads, and when we activate these neurons with the CNO, what we see is a very robust analgesia. So now the cuff animals they tolerate even more force than the control animals. So this is a robust analgesic effect that is driven by the activation of these cells. When we activated, this is again opposite to what we saw when we activated the PKC delta, that we saw a completely opposite phenotype. So taking everything together, what we concluded is that activity of the somatostatin neurons is anti-nociceptive, whereas the activity of the PKC delta neurons is pronoceptive. And if you remember the beginning of my talk, this sort of goes together with the idea that the central amygdala can indeed have a dual function, and that the recruitment of different specific cell types is what's driving or deciding whether the behavior is going to be pronoceptive or anti-nociceptive. So to recap, that was that's all published, and it's the biggest part of the talk. So now everything that I'll show you is the follow-up studies that are ongoing. But basically what we have shown is that the central amygdala functions as this rheostat system, and that increases in the excitability of PKC delta cells drives hyperalgesia or is pronoceptive, whereas the opposite is true for the somatostatin cells, that their activity drives anti-nociception. So what we wanted to know is, now we want to zoom in. We have a nice view of what these cells are doing at the behavioral level. And we wanted to know if there's anything special about the somatostatin or the PKC delta cells, both electrophysiologically and morphologically. So what we did is that we took a step back, and now these animals are naive animals. And because we wanted to know, are these changes because the cells are different to begin with? So what Anisha did is that she patched neurons from either PKC delta or somatostatin cells. This is in baseline levels. And she did a whole array of looking at all the different properties of membrane properties of the cells. And to make a very long story short, the bottom line is that the somatostatin neurons, shown in blue, are more excitable in general than the PKC delta cells. And this is all also supported by all the different parameters that we measured. Now, the neat thing is that what we did is that we filled these cells because we wanted to know, are these neurons also morphologically different? So when we fill the cells, what you're seeing here is a three-dimensional reconstruction of the neurons. And, you know, I remember showing this to my son, he's 12, and I told him, Papi, do you see anything different between these two cells other than the color? And he immediately got what the differences are, which is basically, and I hope everyone can see it too, it's all quantified here. The neurons of the somatostatin, they're more compact, but they have a lot more ramifications than the neurons with PKC delta that have longer dendrites, but a lot less complex ramifications. So that is quantified here with the number of intersections, showing that the somatostatin have a lot more complex dendritic arborization. If we look at the number of dendrites, consistently the somatostatin are higher. But when we look at the length, now the PKC delta cells are the ones that have longer dendrites. Now, we try to tie that back to the electrophysiological properties, and what we did is we used the capacitance, which is the cell size of the neurons. And when we look at the capacitance as a function of the number of spikes, consistent to what we see with our eyes and with the electrophysiology, what we see is that the smaller neurons, which are shown in blue, and the somatostatin, then tend to be, they tend to fire more than the bigger neurons that fire a lot less. So this is showing that there might be a link between the morphology and the electrophysiology. And we're now looking at whether these changes are, there's morphological changes that are associated with the plastic changes that we see in the context of pain. But those are ongoing experiments. Now, in the next few minutes, I'll tell you what we know about the inputs and the outputs. So we know that I started by telling you that the classical pathway that's been characterized is the spino-pontoamygdaloid pathway. But the truth is that the function of this specific pathway, the PB to CEA pathway in the modulation of pain, it hasn't been established. So we have a lot of electrophysiological and anatomical data that sort of points to this being the way that the nociceptive information is being transferred to the central amygdala, but there's not been any causal experiment to demonstrate it. So what we did with that is using a similar trick, but now what we're going to do is also with the Krilox, but in this case, we put a retrograde virus in the central amygdala, and that retrograde virus expresses Cre. So what it will do is that all the neurons that project to the central amygdala, so the terminals, will pick up the virus, and it will be retrogradely transported to any area of the brain, any neuron that sends inputs to the CEA. So now the parabrachial and any other neuron that has projections to the CEA will express Cre. Now, in order to only manipulate the PB neurons that go to the CEA as opposed to the whole brain, then we do a second injection with the DREAD virus that is dependent on Cre, and we put it in the parabrachial neurons. So now what this will do is that only the neurons in the parabrachial that go to the CEA will have the inhibitory DREAD. We do the same behavioral battery of behavior that we have done. Oh, no, actually, this one is different. So in this one, we are using a model of inflammatory pain. We inject an inflammatory agent in the paw, and then we measure the time spent in nociceptive behavior defined as leaking, flinching, and lifting of the injured paw. And in this case, more time spent in nociceptive behavior means more pain, less time, less pain. So as you can see here, when we inhibit the parabrachial to CA pathway with the CNO, activation of the inhibitory red, we see a marked reduction in the nociceptive responses to inflammation. When we poke the animal, the way that we have done before with the cough model, we also see that the inflamed animals have very low thresholds for like respond to very low filaments, but after we inhibit the neurons that project to the CA from the PB, now we have a reversal of the hypersensitivity. The animals can tolerate the force just fine. So this is demonstrating that indeed these glutamatergic inputs from the parabrachial to the CEA contribute to the hypersensitivity that we see in the context of injury. Now, the last few slides will basically cover what we have done for the outputs. And I will focus only on the PKC delta cells because of just to be conscientious about the time. And the idea is to give you a sense of like a flavor of the approaches that we use to address these questions. So what we do is, again, we take advantage of the Krylov system, but now we put a Kry-dependent anterograde tracer in the PKC delta or somatostatin animals. In this case, is the PKC delta. And any cell that expresses Kry will pick up the tracer, which will be anterogradely transported to all the terminals. Then these very heroic two trainees in the lab, Spring is a postdoc, Daniela was a summer student, they take the entire brain, they make thin slices, they mount them, and they go and they search for terminals throughout the entire brain, and they map them. That's not a trivial work. It's a lot of time in the microscope looking at these tiny little terminals that we use morphological criteria to define. So when they did that, we identified a number of different structures, but the one that I will use as just to give you a flavor of what we see is the zona incerta. It's a subthalamic nucleus that's located around here. This is a coronal slice. And the reason that one got our attention is because previous work from Asaf Keller lab at the University of Maryland, they had demonstrated that in the context of pain, the zona incerta is inhibited. The activity of the zona incerta is inhibited. Given that we know that the PKC delta cells are GABAergic, they are inhibitory, and we know that activity of these cells is increased in the context of pain, then we postulated that the source of inhibition for the zona incerta is coming from the PKC delta expressing cells. So we always like to do sanity checks before we go crazy testing the behavioral hypothesis. So the sanity check in this case is that we injected a retrograde virus into the zona incerta of the PKC delta Cre AI9 animals. It's just to verify the injection site. And now what you're seeing is that all the cells that express PKC delta are red, and this tracer will be retrogradely transported to any brain region that projects to the zona incerta. So what we're seeing here is that we see a very robust uptake of the tracer in the central amygdala, and importantly, as you can see in the magnified image and the quantification, a lot of the neurons that pick up the tracer are PKC delta positive, demonstrating that the terminals that we saw and used morphological criteria, this was not just that we were seeing things. This is actually a projection that exists from the central amygdala PKC delta to the zona incerta cells. Now, again, we like to listen to the neurons' response electrophysiologically. So what we did here is that we injected channel rhodopsin, is a light activated ion channel. We injected that in the central amygdala, and we waited for it to be expressed everywhere. And then we make acute slices of the zona incerta. Now, the zona incerta, in theory, if if our hypothesis is correct and our anatomy work is valid, then we should have terminals in the zona incerta that are coming from the central amygdala. When we shine light with the microscope, then we can record whether the neurons in the zona incerta are actually responding to the light stimulation. And what we're seeing here is basically that, yes, we had terminals, we had uptake of the retrograde tracer, and now we're super convinced that this is a functional synapse and it's monosynaptic because we have very robust monosynaptic inhibitory responses, inhibitory postsynaptic responses in the zona incerta cells. Now, behaviorally, and I am almost done. Behaviorally, when we inhibit the zona incerta, now we would expect to have hypersensitivity because that's what we see when Asaf recalls from in his experiments in rats. And what we see is exactly that. So if we inhibit the activity of the zona incerta with the chemogenetic approaches, specifically in the zona incerta, we see that the animals develop bilateral hypersensitivity in spite of not having any tissue injury. Now, the neat thing about this is that these results are identical to the results that we see when we activate the PKC delta cells, supporting the model that these two events are correlated or are linked together. We did the opposite manipulation. So when we activate the neurons in the zona incerta, we want to see, can we bring back the animal now and be no pain? And that's exactly what we see. So now these are all cough animals. And when Suduman injects CNO to activate the neurons and get them to be like that, now the animals are tolerating more force. And again, the neat thing about it is that when we block the PKC delta cells, which would be similar to here, we also see a similar effect. So just to summarize everything that I've told you, the central amygdala functions as this rheostat system, and that is mediated by differential recruitment of cells with the PKC delta neurons having increases in excitability and a pronociceptive function, and the somatostatin neurons having decreases in excitability and an anti-nociceptive function. We have demonstrated that these neurons, to begin with, are very different both electrophysiologically and morphologically, and we have ongoing experiments now to look at whether the morphology changes in the context of pain. And we have identified that at least one of the inputs contributing to hypersensitivity is via the parabrachial nucleus, and one of the outputs that we have identified is the zona incerta for the PKC delta neurons, that when these neurons are activated, they seem to be inhibiting the zona incerta and therefore driving hypersensitivity. So where we want to go from here is basically at every one of these points, we want to zoom in and look at the synaptic and cellular responses of the neurons. And of course, my dream is that we will have a complete map of what the circuitry is for all these different cell types, but more importantly, also for different pain-related behaviors, such as unpleasantness and different affective comorbidities that are typically mediated by the central amygdala. So with that, the most important part is the team that gets all the work done in a very efficient, and it's just a pleasure to have them in the lab and work with them. And with that, I'd be happy to take questions.
Wow, Yarimar, thank you so much. What a wonderful talk. Amazing. I just wanted to ask, I forgot to ask everyone, because we are trying to evaluate the BP Endure seminar series. If everyone could just type in your email in the chat, we will really appreciate it. I know it's kind of late now to ask everyone, but we need your email so we can send you an evaluation form. I just wanted to, before you leave, please do so. Some of you can put more information, but if you have to go right away, just type in your email, please. Now, questions. I have one question, and it's this question says, how about the glia, Dr. Carrasquillo? Are there differences in the glia associated with PKC versus somatostatin cells?
So my understanding is that PKC delta and somatostatin are only expressed by neurons. They're not expressed by glia. Now, there's a lot of interest in the field to try to identify the mechanisms of the modulation of pain by glia in different areas of the pain neuroaxis. In the central amygdala in particular, no one has really looked at modulation of pain by glia. So we don't know the function of glia in this context.
Okay. Then there is another question. What are the therapeutic implications of these results? Do you imagine your work eventually being used to inspire and push for pharmacological therapies to reduce the activity of PKC delta cells, increase activity of the central amygdala somatostatin cells, or other cells in the brain?
Yeah, so that is always a tricky question, right? We always aim at developing tools to improve the quality of life of patients. And right now, as it stands, it's a little bit tricky because I started my talk telling you that there's a lot of parallels between the mechanisms that drive pain and the mechanisms that drive learning and memory, for example. So that makes it a little bit tricky because you can imagine that in order for this to happen, one would need to have a way to access the specific cell types in only the specific area of the brain. So right now, that's a little bit challenging because of this practical reasons in terms of accessing these cells in patients. There's a lot of things that need to happen before one can use these approaches in patients. You know, you have to have the approval for the viral mediated expression of these. I hope my dog doesn't start barking. Yeah.
Don't worry.
So we do have a lot of steps. Now, the part that I do like about the work that we're doing, which is not a direct pharmacological approach or like the typical way that one sees this, but many of the pain patients suffer right now with the idea that they feel that they're not seen, that their pain, people don't believe that they have pain. Because in many patients, there's no measurable pathology. And what I think the research that we're doing does is that it shows that you can have changes at the neural level that are independently of measurable pathology in the periphery. You can still have increases in pain that are driven just by the brain. And I think that from an educational standpoint, that gives the patients some reassurance that it's not that they're crazy and they're making up this pain, but that the brain can actually trigger these alterations in the perception of a particular stimulus. But that's a great question. I mean, we're trying, we're hoping.
So there's another question said, what is the result of GABAergic drugs on these cells?
Ha. So the pharmacology work long time ago has been done by global manipulations. So the bottom line is basically that you get something. Can people still see my monitor?
Yes, we can.
Oh, that many of these different behaviors, when you use pharmacological agents to manipulate GABA receptors, you do get effects effects on all these different behaviors. Depending on whether you do bilateral, unilateral, and the specific context, you can get different effects. But no one has looked at the selective manipulation in a cell type or circuit specific manner. One of the things that I'm particularly interested in is precisely that, to see what happens when we manipulate, because we are assuming that these effects are mediated by GABA, but we haven't demonstrated that that's the case. So that's one of the things that we want to do, but it's a little bit tricky to do these experiments in a cell type specific way. So when you do the global manipulation, whatever is the bigger outcome is the one that's winning right now.
Okay. I want to ask about the interaction between the endocannabinoid system and the GABAergic neurons and your prep. Do you see some link there? Because I know since you are in the alternative medicine division, what is your thoughts on that? What are your thoughts on that?
So that is definitely one of the next steps that I want to evaluate. Is looking at both the endogenous opioid and the cannabinoid system. Mm-hmm. People have not looked at that yet, again, in the context of cell type specificity. We know that these behaviors are also modulated by both the endogenous opioid and the cannabinoid system. But what I really want to know is if so I have this hypothesis in my head based on literature that I've read, but I think that the analgesic effects that we see from the somatostatin neurons, that could be mediated by recruitment of the endogenous opioid or cannabinoid systems, but we haven't done that yet. I always say it's in the back burner of a stove that's outside of the lab right now. But we want to do it.
Then is there any gender differences? Have you looked at male and females, or are we still looking at males?
I heard that question. It's like the public shaming question for me. We are doing everything in males right now, and the reason for that is because in the pain field, it has been shown that typically the behaviors between male and females, the behavioral responses are quite similar, but the mechanisms that are driving it, actually, when someone asked about the microglia, there's evidence. I always get it wrong, and I always need my postdoc to remind me, so I cannot remember which is which, but one of the sexes relies more on microglia, and the other one relies more on like inflammatory mediators or something like that. So it's been shown in the pain field that the mechanisms driving even though the behavioral output is the same or quite similar, the mechanisms driving it is different. So because of that, and my desire to get tenure, we have focused mainly on males so that we can get the mechanistic component out. But this is one of the things that actually my postdoc and I were discussing this morning, that we want to use this COVID time to try to now begin to do a lot of the groundwork on the females. Because the problem is that right now we wouldn't even know if we see the same pattern of activation, if it's the same cells. We have zero idea about the females. So we will probably use the telework time to start doing these basic experiments that require a lot of quantification and things like that, but we don't know. Okay. It's like, shame on me. It's on the list.
But we have one last question. It says, have you or anyone in the pain field done in vivo optogenetic manipulation within the amygdala?
Yes. So yes, a lot. So the results that are seen are dependent on the opto approach that is being used. Actually, we have some ongoing collaboration with the Penso Lab at the NIH that we're doing we're looking at a different input that's not the parabrachium, and the way that we're addressing that is by looking at the photostimulation of the terminals. That's still ongoing, so it's very recent, so I don't know the results, but basically, depending on what manipulation you do, which cell, which side of the brain, then, sorry, how do I make this low? We get different, I'm trying to mute it, but I can't do it.
Don't worry, we understand.
So depending on what, on the cell types, the side of the brain, the behavioral assay, you get many different responses. But yeah.
Okay, but one last one because it's from Dr. Peña. So I have to.
Oh my God, this is like everything is happening today. I'm so sorry.
So can you expand on the competition between PKC delta neurons and somatostatic ones? And what environmental / behavioral factors drive pain modulation to go one way or the other?
Now, that is what's keeping me up at night right now. So, and that is what the work that we're currently addressing is exactly that. So I have ideas of what I think is happening. So I'll try to answer. We haven't addressed it empirically yet. But what we know is that these two cell types, they inhibit each other, so that it's been shown that there's a very strong microcircuitry and interconnectivity of all these cells. So I had it in the talk and I took it out because I wanted to make sure I had the approaches down. But basically, what I think is happening is that, I started the talk, I tried to see if I make sense, but I started the talk telling you of these two different types of responses in the brain, right? And while people typically think about analgesia as something good, the way that I've been thinking about this is that there's a good analgesia, which is the one that is from pain relief. But then there's a bad analgesia that comes from being in a negative affective state. So what we have shown in some preliminary work is that the analgesia driven by activation of the somatostatin cells, it seems to be similar to that analgesia driven by a negative affective state. So what I think is happening is that in the context of stress, fear, or what I call freak out analgesia, I think that the somatostatin neurons are recruited from higher brain areas, like it could be cortical inputs, it could be the BLA, it could be the PVT, but it's in a top-down manner, and the somatostatin cells are being recruited. Whereas in the context of pain, the pathological component of pain, I think that is happening in a bottom-up manner, so that we have that pathway that we have been demonstrating. So we are currently doing a lot of studies looking at, well, currently not, but when COVID lets us go back to the lab, the idea is to look at stress-induced analgesia, to look at unpleasantness, to see the condition-place preference, because, of course, if you get analgesia that is coupled with relief, like if you get rid of this and it's just relief, you should be able to get the animals to prefer that side of the chamber. But if you get analgesia that is driven by stress, the animals are not going to want to go back to that chamber. So we're trying to use all these different assays, behavioral assays and manipulations to try to tease out. But it seems quite clear that the analgesia driven by the PKC delta and the somatostatin is different. So now we have to look at exactly what Peña asked. What are the recruiting factors that are driving one way or the other one in physiological ways? And we're working on that. I have a lot of ideas, but we have to address them with experiments. Did that answer your question?
Yes, it did. Thank you so much, Yarimar. We have to wrap it up. Of course, we could continue talking, but unfortunately, we have to finish the seminar. Once again, Yarimar, such a pleasure seeing you and listening to you and seeing how amazing science are you performing at NIH. And we'll see each other, of course. We'll keep in touch. And we would like to encourage everyone to join us next week when we have Dr. Paul Katima from UCLA, Associate Professor at the Department of Integrative Biology and Physiology, that he's going to talk about circadian rhythms and the molecular underpinnings of sleep. So please join us next week for our seminar. Thank you, Yarimar. Muchísimas gracias, querida. Mucho. And all the best. And thank you everyone for coming, and we'll see each other next week. Thank you. Bye.
Bye. Good seeing you.